Today, I would like to discuss an important topic that intersects with our everyday use of technology and our health. Specifically, the effects of electromagnetic fields, or EMFs, on microglia and the associated health risks. EMFs are ubiquitous in our modern world, emitted by devices such as mobile phones and Wi-Fi routers. Recent studies have raised concerns about their potential impact on our health, particularly regarding neurodegenerative diseases. To begin, let's understand what microglia are. Microglia are a type of glial cell located throughout the brain and spinal cord, acting as the first line of immune defense in the central nervous system, or CNS. They play crucial roles in maintaining homeostasis, clearing debris, and responding to injuries and infections. A significant body of research has focused on how EMF exposure influences microglial behavior. One notable study published in the Journal of Neuroinflammation in 2014 by He et al. Explored the impact of EMFs on microglial cells. The study revealed that EMF exposure can trigger a pro-inflammatory response in microglia, leading to the release of cytokines such as TNF-A, Illinois-6, and IL-1B, which are known to contribute to neuroinflammation. Moreover, EMFs have been shown to impair the phagocytic ability of microglia, which is crucial for clearing pathogens and dead cells from the CNS. This impairment is linked to reduced expression of milk fat globule EGF factor 8, or MFGE8, a protein essential for effective phagocytosis. The study indicated that EMF exposure leads to a significant decrease in MFGE8 levels, thereby reducing the microglia's ability to clear cellular debris. These changes in microglial function are particularly concerning given their implications for neurodegenerative diseases, such as Alzheimer's and Parkinson's. A study in pathophysiology in 2019 by Steffi and colleagues demonstrated that EMF exposure alters the metabolism of proteins like amyloid precursor protein and alpha-synuclein. These proteins are critically involved in the pathogenesis of Alzheimer's and Parkinson's diseases. The study showed that EMFs could shift alpha-synuclein from multimeric to monomeric forms, leading to protein aggregation, a hallmark of these diseases. Additionally, oxidative stress and cytotoxicity induced by EMFs further exacerbate these effects, contributing to neuronal damage. It is also important to highlight findings from the National Toxicology Program, or NTP. The NTP conducted extensive research showing clear evidence that radiofrequency radiation, or RFR, affects glial cells. Their studies found an increased incidence of tumors in glial cells of the heart and brain in rats exposed to RFR. This well-established connection underscores the potential risks of RFR disrupting glial cell function and contributing to long-term health consequences. Interestingly, research has also explored potential protective agents against EMF-induced damage. The study by He et al. Highlighted the role of curcumin, a compound found in turmeric, in mitigating the adverse effects of EMFs on microglia. Curcumin was shown to inhibit the pro-inflammatory pathways, namely NF-κB and STAT3, thereby restoring MFGE8 levels and improving microglial phagocytic function. The implications of these findings are profound. Chronic exposure to EMFs could potentially increase the risk of developing neurodegenerative diseases by disrupting microglial function and promoting neuroinflammation. Given the widespread use of EMF-emitting devices, it is crucial to raise public awareness about these risks and encourage further research into protective measures. Impact on neurodegenerative diseases. Neurodegenerative diseases such as Alzheimer's and Parkinson's are characterized by the accumulation of toxic protein aggregates. A study published in Pathophysiology 2019 by Steffi et al. Demonstrated that EMF exposure alters the metabolism of amyloid precursor protein, APP, and alpha-synuclein, ASyn. These proteins are critically involved in the pathogenesis of Alzheimer's and Parkinson's diseases. EMF exposure was found to shift ASyn from multimeric to monomeric forms, promoting protein aggregation, a hallmark of these diseases. Additionally, oxidative stress and cytotoxicity induced by EMFs exacerbate these effects, contributing to neuronal damage. NTP research on RFR and glial cells. The National Toxicology Program, NTP, conducted extensive research showing clear evidence that radiofrequency radiation, RFR, affects glial cells. Their studies found an increased incidence of tumors in glial cells of the heart and brain in rats exposed to RFR. This well-established connection underscores the potential risks of RFR disrupting glial cell function and contributing to long-term health consequences. RadioBio Initiative, exploring new frontiers. The U.S. military's RadioBio Initiative is exploring the non-thermal effects of RF radiation. This initiative represents a significant investment in understanding how these frequencies interact with biological systems. The goal is to ascertain if electromagnetic signaling between biological systems exists and to define the nature of this communication. This research acknowledges that RF radiation might influence cellular processes and gene expression in ways that are not solely related to heating. Mounting evidence for non-thermal effects. Emerging research suggests that non-ionizing RF radiation can have effects beyond mere heating. For instance, studies have shown changes in DNA methylation patterns and gene expression in cells exposed to RF radiation. These effects occur at radiation levels that do not produce significant heating, thereby challenging the traditional thermal-only paradigm. This evolving understanding suggests that we need to reassess our safety guidelines and protective measures. The analogy to flat Earth beliefs. Refusing to acknowledge the mounting evidence of non-thermal effects of RF radiation can be likened to holding on to outdated scientific beliefs, such as the flat Earth theory or the geocentric model. Just as these ideas were eventually replaced by more accurate understandings, the notion that non-ionizing radiation's only impact is thermal is facing a similar reassessment. Stubbornly clinging to outdated views in the face of new evidence does not align with the principles of scientific inquiry and progress. In conclusion, while EMFs are an integral part of modern technology, their potential health risks, particularly regarding microglial function and neurodegenerative diseases, cannot be ignored. The NTP's findings on RFR and glial cells further emphasize the need for caution. It is essential to continue research in this area and explore interventions like curcumin that may mitigate these risks. By understanding and addressing the impacts of EMFs on our health, we can better navigate the balance between technological advancement and well-being. Thank you for your attention.